Okay, let's start looking at these amino acids and start thinking of them again as monomers that are going to make this polymer of amino acids that we call a protein. Now, a protein is just simply a polymer of amino acids, like we've been talking about polymer, 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 and a polymer is just these monomers of structures that are linked together to form a long chain. And in the case of DNA, they were nucleotides. In the case of polysaccharides, they were sugars. In the case of proteins, they're amino acids. And every amino acid is different, but also they have very common structures. They have a very common structure. In fact, they have 2 or 3 of the main parts are the same, and there's only one part that changes, and that's that R. That R group keeps coming up. Remember, the R group is the variable that changes over and over again. The R group in nucleotides were those nitrogenous bases. They could be C, G, T, or A. In RNA, it was either C, G, U, or A, T substituting for U. But in any case, there was just this unknown that depended on which of those nitrogenous bases you put in there that determined the chemical properties of that particular nucleotide. And therefore, that whole sequence, once the sequence was finished. When you look at amino acids, or rather when you look at proteins, it's those amino acids, that R group in the amino acid, that makes the difference from one protein to the other. Why is one protein one shape, another protein a second shape, and a third protein a third shape? What makes the difference between protein 1, protein 2, and protein 3? Well, the difference is these R groups. The chemical properties of those R groups change the whole structure of the protein and therefore the function of that protein. Now let's take a look at this amino acid structure and try to figure out what it actually is. It's really simple. The amino acid is a really simple structure. It's made up of these 3 parts, and the 3 parts are really just simple again. It's that carboxylic acid, hence the acid part in the amino acid. And that acid part is, again, C, double bond O, single bond O, H. And this structure is a carboxylic acid, one of those functional groups that you had to memorize. The other part of the amino acid that stays pretty much constant is this amino part, hence the word amino in amino acid. And amino group is just basically NH2, no big deal. And again, another functional group that you had to memorize. Hopefully you did. Now the third part that really hasn't changed much between the different polymers that we've been looking at in biomolecules is this dehydration synthesis. Now if you take a look at this red area, it's shading out the OH and the H, that they're going to come together and form this water. Water's going to come out, hence the word dehydration, right? And we've been through this before. You've been tested on it. If you don't know it, you better learn it because you're going to see it again and again. It's dehydration, or the removal of a water molecule. Okay? And it's the removal of water that gives you this dehydration. But now you remove the OH, you remove the H, and you get this water. Well, that leaves you with this nitrogen needing a bond, and this carbon needing a bond, because you got rid of this OH, you got rid of this H, so it leaves this nitrogen without a bond, and then it leaves this nitrogen without a bond, and then it leaves this carbon without a bond. So what's an atom to do? Well, they're going to bond with each other because that's what's left for them to do in order to satisfy our 2 rules of neutrality and a full octet. The carbon's going to bond with the nitrogen, and you're going to form this, what's called very clearly, and you better know it, you better learn it, because it's going to be tested on it. You will be tested on it. It's going to be called a peptide bond. That's the bond between the carbon and the nitrogen. That's why a protein, another word for a protein, is polypeptide. Because a peptide bond is what connects this giant chain of amino acids at every end, on each end of the two of an amino acid that's connecting one amino acid to the other, are these peptide bonds. So here's our dipeptide, just like in with sugars, we had a disaccharide. Now let's take a little closer look at this idea of polypeptides, or really specifically, before we go on to that, I'd like to actually look at these R groups and take a look at what are the different kinds of R groups that can go there and what are their chemical properties. Okay, these R groups are crazy. Like I said, there's about 20 of them. You can count them all. I'm not going to count them. What's really key is that you have each one of these has different properties. For instance, these two are acids and they're very negative. These are all positive in charge, but what's key here is that all of these are charged. Now that's important because you can have proteins that have a lot of these amino acids and therefore it's a negative protein. You can have proteins that have a lot of these amino acids and therefore it's a positive protein. These two proteins can come together and of course they're going to stick to one another because positive proteins and the positive and negative are going to attract. All of these would be, if a protein is made of a lot of these amino acids, it's going to be water soluble. Why is it water soluble? Because it's charged, and we know like dissolves like. We could take a look at these uncharged amino acids. So if a protein is made of a lot of these R groups, then that protein is going to be nonpolar because it's uncharged. The side chains are uncharged. Here we have these are polar, but they're not charged. So these would be water soluble again, but they wouldn't be sticking to one another the way these would be. Then we have these amino acids that are hydrophobic, or in other words, they're nonpolar, right? So these are not going to be, if you have a lot of amino acids in your protein that looks like this, then it's not going to be mixing with water well at all. It would be likely that any part of the amino acid that has this is actually going to be dissolved in the lipid layer, which we'll see later when we talk about the membrane. We'll see that proteins with a lot of these amino acids are actually found in the membrane of the cell. No, you don't have to memorize these amino acids. Later on in your education, you'll have to memorize each amino acid. You'll have to be able to look at it and say, okay, well, these are negative and these are positive. And, that might be important for you later depending on your career. But what's important here is that you know that each amino acid is going to give you a different chemical property. And if you know that, then we can have an intelligent discussion when we start to talk about making proteins and DNA going to RNA going to proteins. So just to review a little bit, here again is this R group. And notice the R groups are kind of like alternating across the chain. The whole thing is a big chain of amino acids. The R group sticking out. Each R group could be different. Maybe this one's polar. Maybe this was charged. Maybe this was nonpolar. And maybe this was nonpolar. So in any case, this is going to determine these two are obviously going to attract one another, and this one will move towards this end, that one will move towards this end. These two will attract one another, so this one will move over here, and that one will move over here. So you'll start to get some kind of shape forming. And as you move down, if you can imagine hundreds, if not thousands of these amino acids, they start to impact one another because they start to pull and push and form all kinds of different shapes. Okay, well, here we have an amino acid sequence. This is, again, the difference. What's the difference between this amino acid and this amino acid? This one being glycine, this one being cysteine, this one being alanine. What's the difference between each of these? It's their R groups. Their R groups are different. They have different chemical properties. Well, what's interesting here, and it's interesting on many levels, is that this cysteine and cysteine, the two cysteines, they have these sulfurs, and they bond to one another. They form what's called a disulfide bridge. And this disulfide bridge, it bends because these two attract one another here the whole protein starts to bend. Now these are small changes. These are monomer in size, and of course, as they have monomers in size, they start to create these little small changes, these bends in the protein. Overall, you're going to end up with a really complicated structure, or one that's bent this way in this protein. Another protein might not have cysteine, or more than one cysteine that's close to one another. So they might not form these disulfide bridges. They might be a little straighter, or they might have more cysteine, which means they'll curl a little more. Another interesting point here is the ends. Now remember, we talked about DNA having direction. We talked about DNA having the 3' end and the 5' end. Well, proteins have directions too. They have a carboxylic acid end, the acid part, the acid side that has a specific chemical composition and set of chemical properties, and you have the amino side. And the amino side has, again, a certain chemical property and a series of qualities that make it different, and that changes. That gives you information again. Now we're back to information. Now proteins don't really relay information the way DNA and RNA do, but they do tend to take shape and function differently because of their chemical properties of their constituent parts. So the carboxylic acid side is going to act in a certain way, and the amino side is going to act in another way. And what's probably more important is that each one of these amino acids, each one has its own chemical property that together they start to paint a picture. They start to create a structure that has a specific function. Now, as we look at this amino acid chain, this polypeptide, you'll notice that here's a side chain that's charged. There's actually 2 of them. Remember, these are what? These are R groups, right? They're R groups. So this is the amino acid side. This is the carboxylic acid side here. This is the amine side, the amino side. So amino acid. Here again is the amino side. There's the carboxylic acid side. Over and over again, these are, you can count them, 1, 2, 3, 4, 5, 6, 7, 8 amino acids, all of them linked together by peptide bonds. It's interesting that these R groups, this one here and this one here and this one here, each of these R groups have a different chemical property. And remember, this one's charged, and these 2 have a negative charge. This one has a positive charge. So maybe this positive charge attracts this negative charge. And what's going to happen if they attract each other? This chain is going to start to bend. These 2 are going to start to be pulled towards it, and it's going to be pulled towards them, and it'll create a loop right here in this protein. So you'll end up with something, maybe, and that'll end up looking something like kind of this. As the positive side is attracted to the negative side, you create this loop in the protein. The sequence of the amino acids going from 1 to 2 to 3 to 4 to 5 to 6 to 7 and going on until the protein ends. Each of these, the 1 position, the 2 position, the 3, the 4, the 5, the 6, and the 7, all those different, the sequence of these amino acids, which one is in what position, we call that the primary structure. It's called primary because it is the structure that comes first. Okay, so it's the primary structure, and that primary structure determines everything else. So which R group comes after which R group in the order that it comes determines the primary structure. That primary structure is going to determine the rest of the structure of that protein. So one more moment, going back and reviewing. Here we have the primary structure of this amino acid. These monomers, these things that we're seeing that are put together one after the other, these amino acids all linked together by these peptide bonds. These things have ends. They have a carboxyl end or C terminus, they have an amino end or N terminus, and they have R groups. And each R group then has a different function, a different property that then determines other things and how this protein is going to function. So as you're reading this, as you're looking at this, and I'm going to pause for a couple seconds, and you guys can pause the video and read this if you like. But there really is a kind of a simple, beautiful, simple structure that's repeated over and over again, and that's this idea of monomers coming together to form polymers, and that these polymers then have functions that are determined. The function of the polymer, the function of the big structure, in the case of a protein, it'll be the shape, a protein with a specific shape that's going to act differently from another protein that'll have a different shape, that'll act differently from another protein that'll have a different shape. Each protein, protein 1, protein 2, protein 3, each of the proteins are the function, the structure and function of these proteins. In other words, what they do, whether they build or take apart or whatever they're doing, is determined by this primary structure, this sequence of R groups, the sequence of the R groups, because the chemical properties of these R groups then tell the protein. I hate the word "tell" because it kind of assumes that the thing is thinking. It's not. These chemical properties of these side groups then start to pull and twist the protein into specific shapes, and these shapes then determine the function of the protein. Okay, so if we look at this and we look at it carefully, we see that each of these amino acids, we see amino acid 1 here that's called methionine, another one called aspartic acid, another one called leucine, and another one called tyrosine. Each of these amino acids, each of them having that structure of amino acid, that carboxylic acid, that amino group, and the R group. See the pink, in this case pink, in this case blue, in this case pink, in this case blue. Each one is a different R group. Then as the R groups, as they come together, remember they're going to have dehydration synthesis. So you're going to see the water's going to leave. You're going to see the water leaving. Remember, the water is going to be this hydrogen and this hydroxide group that's here, okay, that used to be there, that's gone now. But this hydrogen and this OH and this H come together, and they're going to form water. And then, of course, the leftover parts of that are going to end up coming out here and bonding. They're going to make these peptide bonds that you see down at the bottom. All these peptide bonds that connect one amino acid 1 to amino acid 2 to amino acid 3 to amino acid 4. Each amino acid is connected to each other by these peptide bonds. This carbon to the nitrogen bond that carbon used to be connected to a hydroxide group, and the nitrogen used to be connected to a hydrogen. Now you have a polypeptide. You have a bunch of peptide bonds connecting an amino acid, a bunch of amino acids, 4 in this case, and that amino acid has this polypeptide has a carboxyl group at the end of one side and an amino end at the end of the other side. So you have a C terminus, and you have an N terminus, the N, of course, being for the amino and the C for the carboxyl. So, okay, you're looking at here at the primary structure. The order, remember, the primary structure is the order of the amino acids. Now, the order of the amino acids, whether it's serine, glucine, and then glycine, and then some other amino acid, that order is the primary structure. Now, remember what we said earlier, that this primary structure here is going to determine the bending and the pulling. So you're going to start to get a shape. Let's say it's an alpha helix. This is an alpha helix, is the shape that you see here. There's a bunch of shapes you can form. The helix, I think it's one you have to be able to recognize. I mean, after all, you're going to see it again in DNA. But whatever the shape, it could be this loop, or maybe it's a kind of a wiggle, or maybe it's a sheet. They form sheets and they call them beta-pleated sheets. But whatever it is, you're going to have all these different shapes that are going to be bending and twisting because of this primary structure, because of the chemical attraction or repulsion of the different R groups are going to make a specific shape. And then, of course, that is going to lead to you having these different sections. And, of course, you can have this section can have an alpha helix because it has a certain kind of attraction. This serine is pulling towards this glycine that's making it turn. And you'll have this side here, from here to here, will be a alpha helix, and another part of it will just be this wiggly thing. And then over here you'll have a sheet. Kind of another side might form some other shape. But each of these shapes are we call these tertiary structure. In other words, if the secondary structure is this part here, this alpha helix, when you have a bunch of alpha helixes mixed with curlicues, mixed with beta-pleated sheets, then we call that the tertiary structures. In other words, the structures that kind of work into one polypeptide chain, when you have one chain of amino acids all connected one to the other. Now, unfortunately for you, the story does not end there. This is one amino acid chain, one polypeptide chain. So here you have from one end the C terminus all the way to the other end of the N terminus of one amino acid, in this case the blue, okay? So the blue amino acid polypeptide chain is one polypeptide chain. So you have this one polypeptide chain just sitting there, and it's not going to be by itself. It's going to interact with another polypeptide chain. In this case, this polypeptide chain is green. So here's polypeptide chain 1, and over here is polypeptide chain 2. Each is their own molecule in itself, each with its own secondary structure and tertiary structure. So you might have maybe 3 of these alpha helixes, and 4 or 5 of the curlicues, and 6 of the beta-pleated sheets. And the green protein, the green polypeptide, might have maybe 4 alpha helixes and 2 beta-pleated sheets, but whatever, they has their own series of shapes that are together. Well, the 2 of them come together and they form this thing we call, a protein. And a protein can be made of more than one polypeptide chain. It could be made of one, but it could also be made of more than one polypeptide chain. And this protein then has a distinctive function, a distinctive shape, because of the interaction between protein or polypeptide 1 and polypeptide 2. We call that quaternary structure. This may be a little hard to imagine now, but you'll see it later when we talk about other very specific functions, very specific proteins. So let's look at this really interesting thing called hemoglobin. It carries the oxygen in your blood and makes a red blood cell. It's actually at the center of your red blood cell. It actually carries the oxygen. There's several of these hemoglobin polypeptide chains in your red blood cell. Each one carries an oxygen molecule. What's interesting here is that this is actually a demonstration of these polypeptide chains forming that quaternary structure we're just talking about. Here's one polypeptide chain, here's a second polypeptide chain, here's a third polypeptide chain, and here's a fourth polypeptide chain. 4 polypeptide chains that create a very, very specific shape that, along with this molecule called heme and this atom, this ion called iron, right? Iron charged with a +2, all these together are able to carry a single oxygen molecule. It's very important because oxygen is actually pretty dangerous by itself. It can cause a lot of damage if you're not careful. So this oxygen molecule comes in, binds to this hemoglobin chain, this grouping of hemoglobin chains, and this hemoglobin in the red blood cell carries that oxygen molecule to every cell of your body where it changes, where it releases the oxygen. The oxygen can be used for respiration. Now we drew that hemoglobin chain the way that hemoglobin protein the way that we had shown, that I had shown you earlier with the diagramming of amino acid sequences or polypeptides, where you had those curlicues. Well, this is what it would look like, where here you have kind of a very clear alpha helix. Over here you kind of a curlicue. There's a kind of a straight piece over here. There's another curlicue over here. But you'll notice that each of these secondary structures together forming a tertiary structure, when you see them come together and form this kind of pretty awesome little molecule, this amazing oxygen carrying device, where it has these 4 different polypeptides all working together to form this quaternary structure. Now, quaternary, if you don't, sounds like cuatro or 4, quaternary. So you have a quaternary structure that actually determines the function of this hemoglobin to carry oxygen. But what happens when something goes wrong in one of these amino acids? What if we changed one amino acid right there, just one of these hundreds, if not thousands of amino acids? What if we just changed one single amino acid? Could that possibly do something to this whole complicated, mean-looking little polypeptide chain? And then that polypeptide chain, the difference, the change being made there, changing the interaction between all of these polypeptide chains, could that happen? Answer is yes. This one little change, this changing an R group on this particular chain of amino acids, okay, on this beta chain of the amino acid, by changing it from glutamic acid to valine, now you caused all kinds of problems. This chain no longer looks like it used to look. Its shape has changed. Therefore, its function has changed. That change in shape and function changes the entire hemoglobin molecule. So instead of the hemoglobin molecule looking like this three-dimensional construct of the hemoglobin molecule where everything's fine, each chain, this red one, this white one, and this white one down here, all of them working together to be able to carry the oxygen molecule in the center, that no longer is the case. It doesn't work that way anymore. Now, because of that one change in the hemoglobin, now that beautifully rounded red blood cell, instead of looking like this, nice, like a big raft, one of those rafts you ride on the whitewater rides at Kalahari, that beautiful raft-looking red blood cell, instead of flowing through the blood vessels nice and easy, they sickle. They turn into these kind of curvy, sharp little things. They get stuck inside the red bloods, the veins and arteries. They cause a lot of pain. They cause a lot of suffering. And we call that sickle cell. The sickle cell and the anemia that's caused by it, because now these sickled cells don't carry as much oxygen. They don't carry oxygen as well. And not only that, but they stop the circulation of blood to different areas, so that causes anemia. So we call it sickle cell anemia. It's a disease that happens a lot, not just in African Americans. Everybody thinks it happens only in African Americans, but in a lot of Mediterranean populations. And we'll see this disease again and again because it's a really good model to look at all the different kinds of ways that diseases can actually occur. And this happens to be a genetic disease because this is just a substitution in the DNA code that causes a single amino acid to change in the hemoglobin chain in one of the 4 hemoglobin chains, that then causes this horrible deformation of the red blood cell. This would be a normal red blood cell. This is a sickle cell. That sickled cell causes trouble with blood circulation. So you see, it's these shapes and functions, it's the structure and function of these proteins can cause a real serious human issue, a bigger issue outside of just oh wow, it's just an amino acid, it's just like a math problem, or it's just a silly little game that Mendoza wants us to play. No, it's actually something that actually impacts living creatures. It impacts human beings, whole families, because of one amino acid substitution, one change in the primary structure that leads to a change in the secondary structure of that area in the polypeptide, that leads to a change in the whole polypeptide itself, so the tertiary structure, which then impacts how it interacts with other polypeptides, so that it impacts the quaternary structure, and that quaternary structure then impacts the whole cell, the whole structure and function of the cell. And actually, if you look at it carefully, this is a perfect example of structure and it's a perfect example of structure and function. If the cell looks like this, if its structure is this way, then it functions normally. It can flow through the capillaries easily and smoothly, no problem, life is good. If, on the other hand, the structure is deformed, if it's changed, if you change the structure, now you've changed the structure, so its functions change. Now it cannot function the way it normally would have. It can't fit through these tiny little capillaries like the normally structured cell would be. It gets stuck. It causes blood clots, and it's not able to carry the oxygen as well. And all kinds of problems happen. Just because you've changed the structure, you've also changed the function. So you see, structure and function are closely related, very closely related. And you'll see this over and over again in biology. It's one of the biggest ideas in biology. Structure and function are related. And it's not just biology, it's also in chemistry and in physics and in every science that I can think of. If you understand the structure, you understand its function. If you understand its function, that function requires a certain structure. Okay, that's pretty much it. I hope this helped. I know it was long, but I think it was necessary to explain all the little details and concepts, especially this really complicated primary, secondary, tertiary, and quaternary structure. It's the only thing I can think of to say is that if you take it one step at a time, if you really think about it, I think you'll see it's pretty clear how this all works.